Okay, everybody, I am Mr. Auerbach. I will be your science teacher today, and I am here to talk to you about the DNA. And so when we hear about DNA...
Should we continue this from our notes yesterday?
No, I don't think you have to. You can call this a new thing and just put DNA at the top. Right, we've seen this image a lot. It's the double helix DNA, and this is, in fact, the stuff that has all of the information that we need to be us, and anything needs to be alive, from bacteria up to everything. And so today we're going to learn a little bit about how it's structured and a little bit about what some of the different parts of it do.
Do you want us to draw that little squiggly?
I do.
I can draw it well, it doesn't really work.
And so right, so this is called the double helix, right? And that's well, there's two things. Helix because it's a spiral. That's the helix. And then double because there's really two rungs of the ladder here. If you were to untwist this, then you would get just really basically a ladder. And with the rungs just going across like this. Okay, I'm making too many. I'm painting myself into a corner here, but so that's what it looks like when you straighten it out. And somehow in here is all the information that we need for how to make the proteins in our body, how to regulate various biological decisions that the body has to make. And so what is that all about? Well, we're going to learn the structure first. I will tell you this. Speaking of the structure, this part here, what do you call this part of the ladder? I know these are the rungs. What would we call the outside part? The what? Help me out here.
Rail.
The rail. What do we think? Okay, we'll call it the rail. The rail and the rungs. And it turns out...
Are we drawing a ladder as well?
Yes, drawing the ladder. So this is this once you straighten it out. And then the rungs are actually the thing that has the code. And I'll give you a little bit of a warning. There's 4 letters in the code, and we're going to see what that means in a little bit. I'll write them up here. We'll get to them later. A, C, T, and G. And so the actual letters of the code are contained here. There's only 4. Is that enough?
No.
Yes, it turns out it is. Because look, hey, I'm alive. So I guess it's enough, yes.
What does it say on the top of the ladder?
Rungs have code. They have the code.
Rungs?
Rungs. The rungs of the ladder are the parts that go across. Rungs. One of those words. It doesn't sound right once you say it a lot.
And then have code?
They have code, yes. They do.
Have code.
They have
Contain the code.
the code? Is that better? They have the code.
Have the code. Okay.
All of the code. So, is 4 letters enough? Yes, obviously, because we're alive and it works. I would say there's, if you know computers, right? 0 and 1 is all it takes to make all of the computer things you've ever interacted with. 0 and 1 is able to translate into that, and it turns out that 4 letters is enough to translate into all the stuff that makes us be alive. So, before we learn about the actual letters, I just want to tell you what this structure is made of. And so this is like all of the other biological polymers that we talked about, which means that you take a simple unit and then you repeat it a whole bunch of times. And it turns out in this case, in our genome, which is all of our chromosomes, we repeat it about 3 or 4 billion times, spread out amongst different chromosomes. And so the actual unit is, let's take one of these. Turns out that you can actually do this too, because the rungs in the DNA aren't actually connected. the rungs aren't connected. It's actually, we'll get to that part in just a second. It's okay if you do it like that. That's all right, because you're going to see in just a little bit that once you blow it up and look at it larger, that it's actually two different structures that are opposite each other, which is why they call it the double helix. Okay, anyway, if we take this, blow it up a little bit, what it's really made of.
Sarah, what by chance use one of your colorful pens?
I'll put it
You want
inside
us to
there.
just draw this in like the center or something?
Do I want you to draw it in the center?
I don't, how big do you want that?
Big enough. I'll tell you what I'm gonna. Let me draw it, and then you can decide. Okay. All right. So the thing that actually has the code is this thing called a. Well, we're just gonna call it a base. B-A-S-E. It technically is a nitrogenous base because there's a bunch of nitrogen in it. But from here on out, I'm just going to call it a base. And that's there's 4 different bases, and here they are. And that's what goes here, in this spot. And it's made of...
What part is the nitrogenous?
It's the part in the center, the rung right here. This is the actual base here. And it looks, I'll draw one of them. It looks almost like a glucose molecule. I could show you all of the atoms and everything in it. It just doesn't matter, at least to us at this level. It matters a lot to people at other levels. And that is attached by a chemical bond to the rest of the backbone is what it's called. And that's composed of a sugar molecule called ribose. So here's your base. Here's your sugar called ribose. It's technically deoxyribose, but let's not torture ourselves with that right now.
What's it like attached by?
Just like a regular chemical bond. And then it's attached to a phosphate group. I'm just going to draw that as a circle because we've talked about phosphates with ATP, and it's the same thing. And that's pretty much the whole thing. This unit repeats over and over again. And so like this is totally incorrect. All of you people on the internet, I know it's wrong. Just saying. It's almost right. I'm just saying. The haters. The haters. But like you'd have another one of these, and then another one of these, and it goes on, and that's the repeating pattern. Each one of those has a base attached to it, and so forth. And so it's like this, again, and that is the, this is what's called, I'm going to erase this part. This is what's called a nucleotide. So what's on the other side? Another nitrogenous base, right? So if you've got another one, you're going to have another base on the other side. I want to show you something over here. Okay, so, right, what is this? Chromosome. You see that? So these are the chromosomes, right? Here's the centromere holding it together. There's another one over there. And it's zeroing in on this little end over here. And you can see this is the supercoiling, and so it's kind of unwinding. And this is the actual DNA molecule, the double helix. And so again, you see, so just so that you know I wasn't making this all up, you've got this whole thing called a nucleotide, is the nitrogen base here. Here's the opposite one. That's the one that it's connected to, and that's the part we're going to next. Here's your backbone here. Phosphate group, ribose, or deoxyribose. Another phosphate group, another ribose, another phosphate group. So that's the backbone all the way down. The part that actually does something is this. And there's two kinds of these.
Can I close the door by any chance?
Yes, you can.
Thank you.
Right, so here's another view of the nucleotide. And let me, right, nope, won't zoom.
Problem solved.
Alright, and I want to show you there's two kinds of bases, and we'll go back over here and show how these relate to each other in just a second. But there's one shaped like I just sort of just showed you over here, almost shaped like a glucose molecule, except that there's a nitrogen here, and there's a lot of differences actually. There's not another C H2O group there. But anyway, these are called pyrimidines, and I said there's A, T, C, and G. Never mind about this one, we'll talk about that in a second. T and C, thymine and cytosine. I'm going to write all these down for you over here in a second. You don't have to worry about it. I just want to show you that they have two different shapes, and it turns out that that's important. These are the other two, A and G, or adenine and guanine, and they are called purines. The main thing that's different is, obviously, this is two ring structures connected, which you may or may not have seen before, but it's a thing, and here they are. And they're connected, and that's all you have to worry about, is just kind of this shape and this shape. I'm not going to make you draw these, I'm not going to make you memorize these, I'm not going to make you tell me like exactly what's in them, but I do need you to know that there's two different kinds. Okay, and with that, we have, let's see, so we have two purines. Let's see, those are cytosine, letter C.
I don't know where I go. Okay.
Uh-oh. Thymine. And then we have the two other shaped ones, which are purines.
And pyrimidines.
Yes, and
Ribosome connected together.
one of them is adenine, and another one is the guanine.
What's the difference between the first one and the second one?
There are slight differences. If you really want to go, like we could look at it. Here. I'll be right back.
Hey.
Why do they have the U one?
That one's in RNA to give you a look ahead. If you look at cytosine and thymine, you can see there's little bitty differences. Like there's an oxygen here, but a nitrogen here. This is just CH and CH, whereas over here there's a CH3 on this one. This is NH down here, but there's still NH. That's the same. That has a hydrogen attached to it. So there's slight differences. You don't have to know them. I'm back. And so C, T, A, and G. So here is let's call this a C. The other thing you have to know about the nitrogenous bases of DNA, I'm going to put these in a box so they look separate, because they are. And I'm going to draw the other strand over here, is that C only matches with another one. You know how I told you that these two aren't actually connected to each other by a chemical bond? They are attracted to each other, and this is why. So let's see here.
C only matches with C, is that what you're saying?
No, C actually only matches with G. So here's G. And it's because they hydrogen bond to each other, the different parts that stick off of them attract each other. And then A It hasn't been 10 minutes
It
yet.
has. I just
It has?
took the 7 and
Okay,
that's the 9.
all right, fine. And A always matches with T. And that's, notice how it's dotted lines between them because they aren't actually connected. They're only attracted to each other. And if you wonder, how can this molecule stay together if nothing's actually connected? It's because when you multiply this slight attraction by like 100 million plus bases, it actually is very firmly attached. If you want to separate these, you have to bring them to 99 degrees Celsius, which is 1 degree under boiling. And so in a DNA lab, you put the DNA in a water bath, raise it to 99 degrees, and they separate.
Oh, we did that in biomass.
Yeah, right. Oh, yes, ma'am.
I have nothing to say.
Somebody just said my name.
I did. That's
Oh, okay. Were you just like, oh, Mr. Arwag,
Yes.
why are you doing this to us?
I wrote it wrong.
Oh, Mr. Arwag. Anyway, A and T always match each other, C and G always match each other. That turns out to be very important.
And
And this is, this is what happens, like, if I was to put, let's say I had C here, and A here, and this is also separate. If I put the wrong one here, like, if I put a T here, they wouldn't be attracted to each other. In fact, they'd be too close. The T would sort of bend in. And if I put an A and a G together, they actually wouldn't fit together and the whole thing would bulge out. And that's no good.